Hello, everyone. Thank you for joining us today for the latest webinar in our EPIC series on the use of NAMs in risk assessment. This series is being co-organized by the Environmental Protection Agency, PETA Science Consortium International, the Institute for In Vitro Sciences, and the California Department of Pesticide Regulation. I'm Amy Klippinger, president of PETA Science Consortium International, and I'll be co-moderating today's webinar along with several others, including Niladri Bomek, a toxicologist in the Human Health Assessment Branch of the California Department of Pesticide Regulation, Christy Sullivan, the Vice President of Education and Outreach at the Institute for In Vitro Sciences, and Keith Salazar from the EPA Office of Pollution Prevention and Toxics. Please note that the slides and recording from today's webinar will be posted online at this link on your screen shortly after the webinar. All future EPIC webinar materials will also be available at this link as the series continues. We have two speakers today who will be presenting on skin irritation testing using non-animal methods and EPA's new decision framework for dermal hazard assessment of chemicals. First, we'll hear from Hans Raabe from the Institute for In Vitro Sciences, and second from Rachel Brunner from the EPA Office of Pollution Prevention and Toxics. We will host a Q&A session at the end of the second presentation. The webinar is scheduled to go for 1.5 hours, and we may wrap up earlier depending on how long the questions go. Everyone is muted. You can type any questions in the question module of your Zoom toolbar, which is located at the bottom of your screen. Please indicate if your question is directed to a specific speaker. And please do not add questions to the chat. We won't be moderating the chat box. We have enabled closed captions. You can turn this feature on or off according to your preference using the toolbar at the bottom of your screen. Before we start the presentations, our colleagues from the EPA Office of Pollution Prevention and Toxics and the California Department of Pesticide Regulation will share a few comments. Keith, would you please kick us off?
Thanks, Amy. So as many of you probably know, EPA has set a goal of eliminating all mammalian study requests and funding by 2035. So to advance this goal, the Toxics Office is developing science-based frameworks to integrate and prioritize NAMs in TSCA risk assessments and decisions. The new skin irritation framework presented today is the latest deliverable and showcases OPP's collaboration with external experts to reduce and ultimately replace vertebrate animal testing. I'm incredibly proud of this effort and excited for what's ahead. And with that, I'll turn it over to Niladri.
Great, thank you, Keith. And Niladri, please go ahead.
Yeah, hi, Amy and Kit. Hello, everyone. And so on behalf of everyone here at California EPA and Department of Pesticide Regulation, I extend my thanks to all of you for joining today and welcome you for the 7th edition of this EPIC webinar. We at DPR are delighted to be involved with this webinar series from its inception, along with the partners US EPA, IIVS, and PETA Science Consortium. We have a great interest in DPR about this webinar, and many of my peers joined today. To the webinar today, we have a terrific line of two speakers, as Amy mentioned, and I extend my warm welcome on behalf of the panelists here to our speakers today. And we are eagerly waiting for the two presentations on primary dermal irritation and corrosion using the non-animal approach. Perfect timing for this webinar, as just recently we all know about the decision framework came out from US EPA. And we here are eagerly waiting for the presentations, and it will be very important to regulatory agencies like our Human Health Assessment Branch at DPR for skin irritation hazard evaluation. Without taking any time, I hand it over to Amy again.
Great. Thanks, Niladri. Thanks again, Keith. Great. So I will next turn it over to Hans, who will be our first speaker. Hans is Senior Vice President and Chief Operating Officer at the Institute for In Vitro Sciences and has been involved in vitro toxicology assays for more than 35 years. Hans has participated extensively as study director for GLP compliant in vitro bioassays, and as chief operating officer, he is responsible for the overall operations of the contract laboratory services, and he oversees the technical transfer, optimization, and validation of emerging new approach methodologies. Hans has served as an expert on several OECD test guideline panels, has been an invited presenter at various regulatory workshops and review panels, and has participated in multiple ECVAM and ICVAM validation studies. To support the Institute for In Vitro Sciences education and outreach mission, Hans also routinely participates in international in vitro methods technical training workshops. So with that, I'll turn it over to you, Hans. Please go ahead.
Great. Thank you for the introduction, Amy. Let's see, make sure we've got this opened here. And bring it in presentation mode. There we go. I think that you see the full screen there.
Yes, that looks good.
Perfect. Thank you. I appreciate the introduction, and I'm really happy to be able to present here today. Very happy that the EPA has made such great strides in moving forward when bringing non-animal test methods forward. And I'm very happy to be able to give a introduction here on the skin irritation and corrosion test methods, based upon which many of the data in the future will be submitted to the EPA and other regulatory agencies for evaluation of chemicals and potentially products as well. So as an overview for this presentation, we often think about both as data generators and data reviewers. What are the goals of a testing program? Are we looking at evaluating chemicals? Are we looking at the hazards? Can we use tools for more than that? Historically, animals have been used to evaluate for skin irritation, corrosion. Other test methods haven't been developed over the years. Many of the models are simply not great models of human physiology. And so we'll touch base on the human physiology and show some of the differences between human skin and other models used in skin irritation corrosion testing. There's been such a drive to focus on human relevant test systems. And so a key area that we'll focus on here today are some of the test methods based on reconstructed human epidermal tissue models that are available globally. And then lastly, we'll touch base on testing procedures using these tools and a brief on top-down, bottom-up testing strategies. And that should hopefully give a good background for Rachel in her presentation. So moving on. Clearly, skin, our largest organ in our body, is our primary line of defense. And so we need to be able to evaluate for what chemical impacts there may be on the skin. And there are a wide range of endpoints that are looked at. Today, we'll be focusing on skin irritation and corrosion. And some of these exposures could be planned exposures, such as you may have for cosmetics or personal care products, or they may be occupational exposures, such as you see this person in the field working with coveralls and protective gear. We want to understand what the liabilities are to the skin if skin exposures occur. Okay, starting out a bit with the human skin physiology. Laser pointer here. So we're going to focus mostly on basically what happens if test chemicals applied to the skin or exposed to the skin. We've got a barrier function here, the stratum corneum, primary barrier function in this skin model. There are layers here, a lot of complexity in the skin. There's epidermal tissues, which are viable cells, dermal tissues that have a number of different components to it. We're going to focus today on the primary physical barriers against mechanical, chemical, and microbial insults, and focusing mostly on the chemical exposures there. This is a cross-section of human skin. What's really important to notice on here is that we've got 100 micron thick tissue here in the bar. It's about 50, 60, 70 microns thick, depending on where this epidermis here is laying over the dermal tissue. We've got a stratum corneum. That's the primary barrier function. And there's a lot of differentiated cells here, starting from the basal cells that differentiate upwards and display cells and terminally differentiate into the stratum corneum. And before we go on to comparing that human skin model in vivo to some of the various models, I want to cover real quick on this bricks and mortar concept that's been described for the stratum corneum, the barrier function in the skin. And essentially, we've got corneocytes. These are cells that have terminally differentiated. They've They're keratinocytes, epidermal cells that have now keratinized. They're dead cells. They are basically rich with lipids on the outer layer here, keratin granules. They're very protective here. And for a chemical to permeate through this model, there are a couple of different avenues. Small organic chain molecules may permeate readily through and diffuse through the model. Some corrosive chemicals, based on their lytic abilities, could solubilize this stratum corneum as they lyse their way through the tissue into the target epidermal or dermal cells. Some chemicals that may have an affinity for lipid or aqueous environments might just take a more tortuous path, diffusing through this lipid bilayer here between the corneocytes. And we do have to be very careful to understand that each of the mammalian models have different densities of hair follicles. In humans, we have a relatively low hair follicle density, but this becomes a very effective passage route for chemicals to bypass the stratum corneum to get deep into the epidermis and dermis. So we'll keep these concepts in mind as we now look at species comparisons of different mammalian tissue models. So again, here's a sample of human skin, native human skin. And as I showed before, you got a feature of fairly thick epidermis here and relatively robust stratum corneum. And if we compare that against mouse model, which very thin skin model here, minimal tissue thickness in the epidermis and stratum corneum, you can say the same for the rat, maybe a little bit thicker. Rabbit, the same thing. They're relatively sensitive models and have relatively thin barrier functions. And look at these frequent hair follicles here that bypass the stratum corneum. So the human skin is notably more robust in comparison. So as we start talking about some of the models that we can use in vitro to evaluate for skin corrosion and skin irritation, there are several models available in the marketplace. We'll touch base a bit more on this shortly, but I just want to point out the epidermal models have a very similar thickness and structure to epidermal skin in humans. And there's also on the market various full thickness models available as well that include both the epidermis and the dermal compartments. I wanted to point out some brief differences between the rabbit skin and the human skin, since the rabbit skin is used in the test guideline 404 skin irritation and corrosion test methodology. And again, you get a good close-up here. These are relatively similar magnifications. You get a good feel for the robustness of human skin relative to that of the rabbit skin. And there's some metrics here that actually are quite conservative. I think the metrics here that I found in literature show a bit more similarity here than the true differences we really see between human skin, epidermal skin from rabbits. And there's also other differences here. Hair follicle densities, notably different, difference in the structure of the epidermis. And so that really translates into differences in that in vivo model relative to the events that we might see in humans, where we're trying to evaluate for skin irritation and corrosion. So there's a couple of key differences between skin irritation and corrosion, and these are really falling on a continuum of chemical responses. Essentially, the difference is that for corrosive events, we have events that occur that that penetrate deeper than just the epidermis and cause damage down in the dermal fibroblast layer, where significant collagen deposition and scar formation occurs. And these tend to be events that are kind of permanently scarring. With skin irritation, you have more of a milder response, less involvement, more just the epidermal layer. You may just see erythema and edema, and typically are transient and recoverable. And we would like to be able to, for labeling purposes, be able to differentiate between these levels of irritation. By applying sort of a key event basis and adverse outcome pathway, if you will, for skin irritation and corrosion, in common for all chemicals, you can have some sort of molecular initiating event. And for those chemicals that might be irritating and corrosive, you may see some lytic activities such as lipid dissolution and lipid saponification, oxidation, so on and so forth. And this may occur on the outermost stratum corneum as those chemicals are able to cause more damage deeper into the tissue. They may also have the same effects on the epidermal cells, if not the dermal cells and dermal fibroblasts. And consequences of cellular damage, cell stress, cell death may actually translate into tissue damage. And we think about the skin as an organ. Damage to the tissue may result in scar formation and further damage and loss of hydration control in the skin. So if we look at where the DRACE test falls in, we're looking really at the effects at the organ level, at the tissue level, on the skin level here. And this is really a downstream apical event. Most tests, DRACE tests, are conducted very similarly, depending on the regulatory guideline. Typically 1 to 3 rabbits might be tested sequentially just to evaluate whether upon the first animal, well, you may have a potential corrosive event, and we can minimize impacts on more animals. And erythema and eschar formation, as well as edema, are scored on a score from 0 to 4. And really, it's a time basis approach here, so that events that occur relatively rapidly in 3 minutes would be typical for a potential corrosive, out to 4 hours. And if you don't see any events until hours later, days later, those may be more characterized as a skin irritant. And by taking those scores from the evaluation, then you can plug them into various approaches here. The UNGHS score system has essentially a 3-category approach that allows you to look at the mean erythema and edema scores across 24, 48, and 72 hours. The US EPA uses a bit more complex evaluation to look at the erythema and edema, and provides a 4-category system here, going from a corrosive down to slightly irritating. So over many years, there's been a bit of concern about the reproducibility of the animal test, set aside the ethical concerns and issues, or the potential for the test method to be overpredictive, but just simply asking the reliability and reproducibility of the test method. And a lot of data have shown historically that, at the extremes, either at the severe level or the non-irritating level, most tests can be reasonably reproducible. And so an evaluation of data submitted both to ECHA as well as later in the EPA category system, studies were evaluated looking at hundreds of different chemicals that may have been tested at least one time, if not 2 times, at least 2 times in submissions to the regulatory agencies. One could evaluate if you pulled at random a chemical with a result and got a category 1 result, if you could pull a second study with that same chemical to evaluate the likelihood for getting the same result for category 1 materials, so the corrosives, there is a very high likelihood of getting that same result again in subsequent studies. And very similarly, at the other end of the scale, you see the same type of thing, that for non-categorized materials under the GHS, you may have a very high likelihood for receiving the same classification when tested multiple times. And the challenge here is that in the middle ground, for category 2 and category 3, moderate and mild irritants, there's really quite a bit of variability here in the results. For example, for category 2 substances on the first evaluation, if you got a category 2 substance, subsequent studies for that same chemical may have a notable difference in the category 3 or non-categorized result upon repeat testing. And so category 2 substance that could be a moderate irritant could also be readily identified in subsequent tests as non-irritating at all. And with category 3 substances, you see almost an equal number of times that you see non-category 3 outcomes, both as category 2 or non-categorized. So there's just a lot of noise in the test method in this middle ground. And so that certainly is a concern, both to industry and regulatory agencies. So what can we do about that? What tools can we use in this prediction of skin irritation corrosion that would perhaps we look upstream at cellular responses? And so we've got a number of in-chemico tools to look at corrosivity that evaluate these molecular initiating events on the stratum corneum. We can look at cellular responses, and we can look at tissue level responses with reconstructed tissue models using human relevant cells. And it's both the Crostex and these 3-dimensional tissue models that we'll touch on next here. Okay, so the first test method that actually was one of the first methods to ever be validated for non-animal testing for skin irritation and corrosion is a Crostex test method, and this is called the membrane barrier test method. The method uses an artificial membrane, essentially using a membrane that's constructed of proteins and lipids, fairly similar in composition, if you will, to stratum corneum as sort of surrogate target for the stratum corneum. And the goal here is to build a model that has a barrier that if you apply test chemical over time, you can look at the time for that barrier to be breached and for that chemical to damage that barrier and then breach through. And so we do essentially a time to toxicity approach here. And for chemicals that dissolve that barrier rapidly and break through rapidly, those are likely to be corrosive. And this test method actually allows you to look at corrosives and determine UN packing group subgroups, as well as the DOT packing group subgroups. And this method is one of the first methods to be adopted then in the test guideline program is 435. There are some considerations. There's a chemical detection system that the chemical that you're applying to this biobarrier on top needs to be able to be detected, and colorimetrically use a litmus solution here. So if you have a chemical that changes pH radically from this yellow, you'll see a red coloration, and that'll be your cue that a chemical is broken through. There's also some other considerations to categorize materials based on their high or low buffering capability. And during the run of the test, you apply test chemicals at the top of this biobarrier. The barrier is inside this black membrane here. And if you see chemicals start breaking through, you might see this little red sliver here. And that'll be your first indication that you'll record the time for that breakthrough to occur. So in running this protocol, you basically prepare the matrix powder, comes with the kit. You prepare these biobarrier matrices and add them to the solution. You can solidify it at a refrigerated temperature. And then to initiate the test, you apply the biobarrier on top of this chemical dilution system here, or chemical detection system, and you monitor. You dose up to 4 of these treatments here, and you monitor for the breakthrough time. And this shows after a certain amount of breakthrough time that chemical is broken through and causes color change. And that is your indication of the breakthrough time that's occurred for that chemical. And the breakthrough time is then used to categorize whether a material might be corrosive or not, and if corrosive, what the packing group is. And so if you look at this category here, a breakthrough time that you detected in less than 3 minutes would be a packing group 1. Up to an hour might be a packing group 2, and greater than 4 hours, for example, would not be corrosive at all. The test method has got a limited domain for chemicals that can be detected in this chemical detection system here, but it's got a nice balance of sensitivity and specificity for those materials that are detected to be corrosive. And so next is to look at the reconstructed human epidermal test methods. And these are human relevant test systems. And the first area we'll look at is skin corrosion. Here's a graphic showing what the tissue inserts look like. They're cultured at air-liquid interface, so they're placed inside media. On the top side is at air exposure, just as our native skin is. This histology cross-section shows the proliferative basal cells, granular cells, stratum corneum, and these would be bathed by media from below, just as our skin is bathed by the circulatory system immediately below the epidermal level. And there are quite a few different tissue models here. They all have a lot of characteristics in common. They all are based on human viable keratinocytes that are seated on a basement membrane, and they proliferate, they differentiate. They have distinct differentiators, and they all have a stratum corneum. And this provides a barrier function for testing chemicals. The EpiSkin model has been discontinued, but I still include it here because it's very possible that existing data are still available and may be submitted for regulatory review based on the EpiSkin model. So I think it's important to remember that this model, the data from this model are still potentially available. There are models available from Germany, from France, from Europe, Japan, Korea. So there's quite a few models available globally. So going back to this time to toxicity concept, this concept is really based on that if a chemical causes damage rapidly, penetrates the skin, it lyses skin, and causes damage rapidly, it is likely to be corrosive or irritant. And for those chemicals that might be applied to skin and well tolerated for hours on end, if not days, we think of those chemicals to be non-irritating or non-corrosive, certainly. And so there's this time-related continuum that different chemicals may fall under. And there's some representation of different products and chemical classes that might fall into this continuum. And the consequence here, of course, could be that you may have corrosive events on skin for those materials that are corrosive. And ideally, we're looking at, for personal care and products, to have those materials that are not irritating. And this time to toxicity concept can also be applied in vitro. We can utilize these three-dimensional tissue constructs to look at whether material might be tolerated for very long exposure times, or if they kill tissues rapidly, those might be typically of corrosive materials. And Mattek, who is a manufacturer here in the States, has come up with some guidance here for the exposure time that could cause a 50% reduction in viability, and how those different chemicals and their irritation potential might fall in based on their ET50 values. So the basic protocols allow us to look at these reconstructed human epidermal models by looking at the viability of the test system after exposures. And viability is typically done by looking at placing the tissues after exposure into a MTT solution. Typically, that MTT is a yellow color. Mitochondrial reductases through respiratory activity will reduce that yellow dye to a purple formazan, and we can look at that purple coloration in these tissue models, extract that formazan out of the tissues at the end of the study, and basically look at the viability spectrophotometrically. So basically, this is what the tissues look like upon receipt. We can dose the tissues topically, just like may happen in vivo. We can rinse the tissues. We conduct that MTT reduction assay, and we can extract the MTT and get objective data using a plate reader here to get data on each tissue. And we can take those data then and plug them into a prediction model. If we look at the treated group versus negative controls, then we can get a relative viability value. And this series of prediction models shows for each of the commercially available tissues, there are essentially very similar prediction models here that determine whether material might be corrosive or non-corrosive. And there's also the ability to do some subcategorization. And again, as I mentioned, the EpiSkin model has been discontinued commercially from availability, but I'm sure there's still plenty of data that are in the public domain for which that may be used for submissions. And so I think it's important for us to bear in mind this prediction model as well. Okay, there are also some key caveats that need to be made aware for reviewers. There's potential for chemicals to be direct reducers, so we need to use freeze-killed tissues there. That should all be in a study report. Similarly, if a chemical causes coloration that is persistent through the assay, one can have controls to address that. There are other parameters here on how to dose the materials. And so these type of details should be captured in study notes. Then moving from corrosion to skin irritation, this test method also is approved on the OECD test guidelines program and looks at the milder spectrum for irritation. There are also some specific notes here to consider for conducting these studies. A mesh is used to ensure that the low volume applied to the tissue can spread through capillary action across the top of the tissue. So we want to make sure that we don't have any potential interference with these meshes by the chemical alone. So we do some pre-testing there. So the test approach for the skin irritation is also very similar to the corrosion assay. Again, we got the same tissues upon receipt. Treatments tend to be a little bit longer, whereas in the corrosion assay, the treatments may be in the 3-minute to 1-hour time frame. Here, the skin irritation test allows you to apply test chemical for a bit longer exposure time. With the epiderm model, it's up to an hour. But you also have this post-treatment incubation for expression of potential irritation effects to occur. And this expression time can be out to 42 hours. And then we conduct this cell viability assay again, which is, again, a hallmark of skin irritation is the loss of cell viability. And we can read those values spectrophotometrically. And this brings us again to a prediction model. If we see that the tissues have, if the chemicals cause a reduction in viability of less than 50%, then we would classify that material as an irritant. And conversely, for tissues that are well tolerated, chemicals are well tolerated on the tissues and viability is greater than 50%, we would categorize this as a non-irritant. And for these gives the UNGHS categorizations. Bear in mind here, and this is something that will come up also in Rachel's presentation, is that for the skin irritation test, if you get a positive prediction, it could be a category 1 material as well, not just necessarily category 2. So one needs to identify whether the material might also be corrosive. So you may need to go then from the skin irritation test to a corrosion assay, which brings us then to this IATA for skin irritation and corrosion potential. And so the top part of the IATA really has you look at what available data might be available in vivo, in vitro, and so on and so forth on your chemical of interest. If you need to proceed to testing, then you would look at a top-down or bottom-up approach here using these test systems. And within this testing paradigm, if you come in with a top-down approach, expect the material to be corrosive or irritating, you might start with the corrosion assay, either the Crostex or the human relevant epidermal corrosion assay. And if you get a corrosive result, you can label it that way. If you get a negative result, then you need to go on to the skin irritation test and determine if it may also be potentially a skin irritant. And that can subcategorize into irritant or essentially non-irritant, mild irritant. And if you start with the bottom-up approach, conversely, you might start off with a skin irritation test. And if it comes up negative, you're done. You've got a non-irritant prediction in GHS categorization scheme. If you get a positive prediction here, as I mentioned earlier, then you also need to look at the skin corrosion test methods to determine if it may just simply be an irritant or potentially also a corrosive. Okay, so I hope this gives you a launching point here then, some basis as Rachel then proceeds with the presentation on the testing framework. Go ahead and stop sharing. Thank you.
Thank you, Hans. Really great presentation. And I see we already have a couple questions in the question module, but please continue to add them as you have questions. You can put them there at any time, and do note if they're for a specific speaker. All right, so following Hans, next we'll hear from our second speaker, Dr. Rachel Brunner. Rachel studied molecular, cell, and developmental biology at the University of Illinois at Chicago and completed a PhD in biological sciences focused on the intersection of heart development, disease, regeneration, and epigenetics. Rachel joined EPA as an ORISE postdoctoral fellow, where she modeled key aspects of heart development using human iPSC-derived cardiac progenitor cells. Rachel then went on to join EPA's Office of Pesticide Programs and worked on projects in support of risk assessment of pesticides, such as incorporating toxicokinetic considerations to set more human-relevant dosing, and worked to reduce testing in dogs. For the past 4 years, Rachel has been a human health assessor in EPA's New Chemicals Division of EPA's Office of Pollution Prevention and Toxics, assessing new chemicals regulated under the Toxic Substances Control Act. Alongside risk assessment, Rachel has also pursued increased use of NAMs in this work, and she will provide an example of that today. Go ahead, Rachel, your slides look good.
I had to figure out how to unmute myself. Thank you, Amy. That was a great introduction. And thank you to Hans. That was a really great setup to talk about the skin irritation and corrosion framework that we just published. So thank you for that. So today I am going to talk about the implementation of this NAMS-based framework to evaluate skin irritation and corrosion potential of new chemicals submitted under TSCA. Like I said, this is recently published, if you'd like. We have a QR code right here if you'd like to follow along. Before we get going, just a disclaimer, the views expressed in this presentation are those of the authors and do not necessarily represent the views or policies of the US EPA. Any mention of trade names, manufacturers, or products does not imply an endorsement by the US government or EPA. And EPA and its employees do not endorse any commercial products, services, or enterprises. Okay, so who are we and what are we doing? So new chemicals were housed under the Office of Chemical Safety and Pollution Prevention, or OCSPP. Within that office is the Office of Pollution Prevention and Toxics, or OPPT, and the New Chemicals Division is housed within that office. And NCD is responsible for making a safety determination for new chemicals submitted under the Toxic Substances Control Act, or TSCA, prior to entry into US commerce. And as part of this process, we assess for the potential for skin irritation and corrosion. A little bit about new chemicals. we say new chemicals, we're referring primarily to industrial chemicals, so not pesticides, not food additives. These are more industrial chemicals. We receive about 500 submissions per year. We have relatively short timelines, 30 or 60 days, to turn these around. Because of that, these are screening level assessments. And to make things more interesting, there are no data requirements for new chemicals, so they frequently come in without any data, including data for skin irritation or corrosion potential. So we employ a number of read-across methods in our program. Our skin irritation corrosion categories that we use in the new chemicals program is a little bit streamlined. We have 3 categories. They're non-irritating, irritating, and corrosive. Okay. So in Hans's slides, he provided a great introduction to this. There are a number of limitations with the rabbit study, both in terms of relevance to human skin, in terms of anatomy and physiology. In addition, he talked about how the rabbit study lacks reproducibility, particularly in the mild to moderate range. And we also talked about the advantages of NAMs. So there are a number of different NAMs available, including those based on human cell-based tissue models, which is fantastic. And they've been found to perform as well or better than the in vivo skin irritation test. And that really gets us to the key principle of the skin irritation framework is using those methods first that are reproducible and relevant to human skin irritation and corrosion potential when doing our assessment. I have a QR code here again, if you'd like, if you missed it the first time. In our framework, the first type of test data that we're prioritizing are those human cell-based tissue NAMs. It's like the EPI-Skin model. If, using this data, or, maybe it's not available or there are data gaps, then we'll consider other types of NAMs like the Corositex assay, that barrier method, the in-chemical method. If we don't have either of these, or there are still questions or data gaps, then we will look to in vivo test methods. But we have a number of criteria. We have things that we think about before we'll use the in vivo methods. Before getting into the details of our framework, in addition to sort of their ethical concerns with using animal models and prioritizing NAMs that have been shown to be more reproducible and have greater biological relevance to human skin irritation and corrosion compared to animals. This framework supports EPA's very recent recommitment to reducing animal testing and eliminating mammalian testing by 2035. This is consistent with the strategic plan also to promote the development and implementation of alternative test methods within the TSCA program. This was developed in response by EPA in 2018 in response to TSCA Section 4H2A, which gets at, the goal to replace vertebrate animal testing and provide information of equivalent or better scientific quality, which I think we can do here, and relevance for assessing risks of injury to health or the environment. In addition, the skin irritation and corrosion framework fulfills in part the long-term goal of the strategic plan. This is important to integrate and prioritize NAMs in the TSCA decision process for risk evaluation, to reduce and eventually replace the use of vertebrate animal testing. Just realized you can't see me. Here we go. Okay. now we're getting it into this a little bit. So as Hans showed, the NAMS assays are binary. The outcomes are binary. So we have three categories, right? Non-irritating, irritating, and corrosive. But currently there are no NAMs that can distinguish across these three categories. There's two types of binary assays. There are a type of binary assay that can identify things that are not irritating. But if it's not non-irritating, it's either irritating or corrosive. But it can't distinguish between these two. There are also binary assays that can identify corrosives. But if it's not identified as corrosive, we don't know whether it's non-irritating or irritating. These were presented in Hans' slide as sort of the top-down and bottom-up approaches in the IETA. To identify irritants, you need to use both kinds of assays. I have the OECD 439 and 431 listed just as examples here. And if you have both kinds of assays and it's a non-irritant in the assay that can identify non-irritants and it's not corrosive in the assay that can identify corrosives, then you can definitively say that something is irritating. And Hans went over a number of assays that are available for this. They have there's a number with OECD test guidelines, which is fantastic. We'll also consider others on a case-by-case basis. The OECD test guideline 439 for in vitro skin irritation. This is a human cell-based tissue model. This one can identify non-irritants, but again, can't distinguish between irritants and corrosives. And then there's the in vitro skin corrosion test, or OECD test guideline 431. It's a number of models available for this one and can identify corrosives, but again, can't distinguish between non-irritants and irritants. An example of another NAM that's not a human cell-based tissue model is the Corositex. Well, Corositex is the test method, but the OECD test guideline 435. So this is an in-chemical method that can test for corrosives. And I think there was a question in Q&A about whether, this assay can't identify if there's like going to be reversible damage. This would be like irreversible damage. You would need another assay, such as the 439, to determine if something is an irritant. Okay, so I'm going to show this in a little bit of a different way because this is how we present it in the framework. So this is a little bit, this is part of sort of our framework diagram. So the two orange boxes, this one and this one, represent the two different kinds of binary NAMS assays. So you have the kind that can identify non-irritants versus we're not sure if it's irritating or corrosive, and the ones that can identify if something's corrosive, but can't discriminate between irritating and non-irritating. So in this one, if it's non-irritating, okay, you can classify it as non-irritating. In this assay, if something's corrosive, okay, you can classify something as corrosive. I haven't actually seen it come up yet where we've had these assays and one said non-irritating and one said corrosive for the same substance. So that hasn't been an issue yet. And then you can combine, in order to get to irritating, you can combine results from both assays. So to get to that classification. All right, so now we are going to combine this with the prioritization I showed earlier and a little bit about, new chemicals program tends to be data poor. So we'll start with that part. So we start up here in the left. This is our decision framework. First, we ask ourselves, do we have skin irritation or corrosion data on the new chemical substance itself? Frequently, we don't. Or maybe we have like a little bit, maybe we have like a 439, but it doesn't really give us a definitive answer. We are going to look to similar substances, usually structural analogs, to see if there's data available for those. And once we sort of collect our data, we will prioritize that data based on whether it's a human cell-based tissue NAM, whether it's another kind of NAM, or if it's in vivo data. We're going to look at the human cell-based tissue model NAMs data first. And if we can, and we'll go over to this gray box and we will look at that data and see if we can get to a classification using it. If we cannot, or if that data doesn't exist, what we'll do is we'll consider other NAMs data to address any data gaps in this data or the lack of this data. We'll go back into this gray box and kind of, if we have data from both types of assays, we'll consider them together. So let's say, maybe we have a human cell-based tissue model NAM, like a 439 here, and it says like irritating, corrosive. We can't really tell from that, but then we come down here and we have a NAM that's like this, and it comes out as irritating, non-irritating. Then we have both pieces to say that something is irritating. But maybe we don't have this information, or maybe there's a data gap. At that point, we'll consider whether or not to use in vivo data. So maybe we have some data here and we have in vivo data, but it's not the only data available. Most likely, we're going to use the most severe category from the NAMS data. So let's say, we have a binary assay here and that's it, and it's either irritating or corrosive. Even if we have in vivo test data, in most cases, we're going to select the most severe rating. So we would classify it as corrosive. In limited cases, perhaps we have a situation where we have, 5 in vivo data, 5 in vivo studies on our structural analogs, and they're good structural analogs. And all of those studies are saying the same thing. And let's say, as an example, they're all saying that the substance is irritating to rabbit skin. In that case, what we're seeing in the in vivo data is very consistent, and we might, instead of saying corrosive, we would say irritating. Another situation, maybe, there's no data here, all we have is in vivo data. If that's the case, we'll use it. And then similarly, if there are no in vivo data, we have some information up here, we will just use the most severe category, like I described earlier. Okay, so I just said a lot here. So I put together some examples, starting with some maybe easier ones. So on the top here, I'm showing okay, we have these 3 data points. We have either on the new chemical substance or close structural analogs. We won't get into that part. And we have an OECD 439. This is a human cell-based tissue model. So this is our highest priority, showing that this substance is not irritating. We have a 435. This is another kind of NAM showing it's either non-irritating or irritating. And we've got an in vivo study saying it's not irritating. Okay. So we're going to look at this data first. And that gets us into here. It's not irritating. And we would classify it as non-irritating. We wouldn't necessarily even look at the 435 and the 404, or we wouldn't consider them. In this case, they're consistent with this call. Okay, how about another example? Let's say we have an OECD 439, it's a human cell-based tissue model, and the result is irritating or corrosive. Okay. So that brings us here. It's either irritating or corrosive. And we also have an OECD 431, which is this type of binary assay. And it comes out as non-irritating or irritating. Since we have both of those, we can classify it as irritating. All right. Got another example. Okay. I've already forgotten my examples. Okay, so in vitro skin irritation, we've got an OECD 439, again, a human cell-based model. The result is irritating or corrosive, so that gets us here. We have an OECD 435. All right, so we're here, but we have now run out of human cell-based data. So we're going to come back out. We're going to look to other NAMS data and come back in here. We know we're still here. And this data, the other NAMS data, suggests that it's corrosive. In this case, we would classify the substance as corrosive. There is this in vivo data here that says that it's irritating. However, we know that the in vivo studies aren't particularly reproducible. So we would not use this. We would classify the substance as corrosive. Okay, example number 4. We only have in vivo data. Okay, well, we don't have data here. We don't have data here. We only have in vivo data. Are in vivo data the only data available? Yes, we would just pick the most severe category from the in vivo test, which is irritating. And I'm happy to go over other examples as well. There's so many possibilities. So just in conclusion, this framework prioritizes reproducible and human relevant data for assessing skin irritation corrosion potential. We really think it's going to encourage new chemical submitters to provide more NAMS data, which in turn will result in reduced vertebrate animal testing, which would be fantastic. It also improves the efficiency, consistency, and transparency because we have a process, we have an external framework that anybody can look at. And this is really going to help with the assessment of skin irritation and corrosion potential within the new chemicals program. And it will also serve as a resource for stakeholders preparing new chemical submissions to EPA under TSCA. With that, thank you to everybody who helped with this effort at EPA, Renee Beardsley, Keith Salazar, and Anna Lowitz. The Institute for In Vitro Sciences, Hans Raabe, Amelia Costin, the PETA Science Consortium International, Amy Klippinger, Anna Vanderzalm, and Adam Bettman. Thank you guys so much. We could not have done this without you. And just a plug, just this work is just part of a growing landscape of NAMS work. In EPA OPPT, the New Chemicals Program. We have used NAMS for read across for a long time. We use analog data, both from sort of our internal databases and publicly available databases, category assessments from national and international hazard and risk assessment entities. Many of you might be familiar with our New Chemicals Program, chemical categories. We use structural alerts. We use in silico tools such as Oncologic and the OECD QSAR toolbox. And we're trying to add more NAMS to this process. This is just part of that. And this is actually very similar to the somewhat recently released decision framework for eye irritation and corrosion. So when you read this, you'll see it's very similar. That Renee Beardsley had champions. There's also, we were part of an upcoming respiratory sensitization OECD detailed review paper. Also up and coming is a framework for read across using EPA's cheminformatics modules. So this is a way to systematically search publicly available databases for read across. In our program. We're also working on the threshold for toxicological concern approach as well. And for these bottom two, they will be, just another plug, they will be presented at SOT Monday morning if you'd like to learn more. And if you'd like to learn more about skin irritation, this skin irritation framework that I presented today, I'll have a poster at SOT as well. And with that, here are the references. And I believe these slides will be posted. Thank you.
Great. Thank you, Rachel, for that really clear, great presentation. We'll have Hans come in and we'll start answering some of these questions. So thank you all very much, first of all, for putting your questions in the Q&A. If you have put a question in the chat, we haven't seen it. So if you could transfer it over to the Q&A, where you can also upvote questions that people have already submitted. So feel free to check that out. We have quite a few questions to get started, so we'll do that. First one is directly for Rachel. Will the EPA skin irritation framework also apply to substances regulated under FIFRA? Can you say anything about that?
This is specifically for new chemicals, and the Office of Pesticide Programs would have to speak to substances regulated under FIFRA.
Okay, thank you. The next question is, if a chemical has a very low pH value, less than 2, a very high pH value, more than 11, is it classified without any testing?
I can take that one. Yes. Yeah. If there is testing and it shows otherwise, we would consider that information. But if it has a very low pH or a very high pH, it would be considered corrosive.
Okay, thank you. The next question is about a toxicity to the skin itself. So it's a bit of a long question, but if I'm paraphrasing, they're asking if existing models, for example, full thickness human reconstituted epithelium, would allow for irritation evaluation of in vitro skin exposed to the chemicals from within the layers and not just the top layers.
Yeah, so it sounds as if the question would be if one could do sort of intradermal type exposure. Is that correct? Is that the way it's read? Okay. I can envision preparing protocols to do that. There are also approaches that if the concern is that the stratum corneum is a strong barrier function, a scientist could take a model that has an immature barrier function or no barrier function. A ocular model, for example, would be essentially the skin model without the barrier function, and could do some comparative studies there as well, and essentially basically bypassing the stratum corneum to look at potential for toxicity to the tissue. There's no regulatory approval of a method like that, but certainly that is something one could model in the laboratory.
Okay, thanks, Hans. The next question is, has in silico modeling been considered as a possibility to determine dermal irritation or corrosion, or as an addition to the IATA? So maybe they're asking both about EPA's strategy, but also the IATA that you presented, Hans.
Not my area of expertise, but yeah, there are in silico models that look at various parameters. They can look at the log p values, look at the pH, for example, and just take some phys-chem properties and make some determinations on the likelihood for skin irritation or corrosion. I think it's probably more of a hazard approach rather than a necessarily risk type formulation or a continuous formulation. Yeah.
Okay.
Yeah, for EPA, typically we have enough test data. Either maybe we have it on the new chemical substance itself, or we can identify something on a structural analog, and we will use that. I think it would be very rare for us to use in silico predictions, but welcome the development of those.
Great, thank you. So there's a question that's come in that is not really relevant to EPA purview. They're asking about veterinary products And is there a model for companion animals? And I don't know, Hans, if you are aware of anything like that.
I'm not. I think for human health effects, where reconstructed models have been developed using non-human systems, some early work was done using rodent or rabbit cell models to recreate a in vitro three-dimensional construct for rabbit, but I'm not aware of models being developed commercially for veterinary application.
Okay, thank you. Okay, so there's a question. First, it mentions that you can classify a compound as irritating, which can be either irritating or corrosive. But can a compound be classified as corrosive but not irritating?
Yes, we pick one.
Right, because you would pick the more severe
Mm-hmm.
classification, right? And they go on to mention that is it based on an inflammatory response like irritation might be, and corrosiveness is based on breaking the physical barrier. So that actually comes to another question that Rachel, you picked up on in your discussion, but I wanted to come back to it because it brings us this question of you know determining the reversibility of chemical damage. And actually, that's really the definition of a corrosive acting chemical is something that isn't reversible, whereas irritation is. And I just wanted to give you both a chance to speak to the idea that, even if previously with the rabbit in vivo model, we're able to understand both of those responses, we can combine, as you've been speaking to, we can combine both different kinds of methods in order to give us a full complement of the potential effects on the skin.
Rachel, would you like to start?
I mean, yeah, I understand like where this is coming from because the readouts, like particularly for like the 431 and the 429, 439, 431 and 4, I can't get them straight anymore, are cytotoxicity. So it doesn't really get at the reversibility question, but I'm going to defer to Hans to talk about that a little bit more.
Yeah. I guess in terms of being able to so there's some applications where one could demonstrate wound healing with these models, but I don't think that's what we're really getting at here. Demonstrating reversibility is something that hasn't necessarily been done from a corrosion level. But I think the difference here is that with the animal test, we've got basically an apical outcome, downstream event. And what we don't necessarily get is the depth of that injury into the epidermis or the dermis. By using these models, we do get a measure of the depth of injury into the epidermis as well as time related. Theoretically, with a full thickness model, dermis and epidermis combined, those type models, one could potentially also do a depth of injury into that model to look at the likelihood of the dermis being involved as well. Sort of by definition, if you've involved the dermis, and again, this is a degree of how much involvement in the dermis occurs, you're going to have a greater likelihood for lack of recovery or some sort of scar formation. I think anytime you have a corrosion event, the corrosive effect is basically under that area of exposure, but distally from that corrosion site, you're also going to have areas of lesser damage and maybe just epidermal only damage, irritation. And that wound healing is going to essentially come from the outer distal ends to cover again that site of exposure. Yeah, I think in a long story made short, I think you can get some decent predictions on the likelihood for recovery by applying the skin irritation and skin corrosion assays, which is a little bit different than looking at the animal test where you're basically evaluating over time to see if a recovery occurs. If you could get that early indication of a depth of injury in the animal test, you would have a good correlation then to the likelihood for recovery.
Okay, thanks very much, both of you. Another question that's coming in is about, and it's directed to Rachel, if there is an in vitro result that shows non-irritant properties, but an in vivo result shows irritation, would you classify according to the in vivo result as an irritant?
Oh, we would classify in alignment with the NAM result, and we would call it a non-irritant.
Okay, thank you. And this might be a question that both of you can speak to. Can you apply this approach to mixtures?
From my perspective, and this is not a regulatory answer, but more just a test method basis, the closer we build these models to human experience, human tissue experience, the more likelihood we'll be able to get accurate predictions. And so with the three-dimensional tissue constructs, you're able to apply test formulations and test chemicals onto the top of the tissue, exactly as happens in vivo. So not only mixtures, which might be simple mixtures of chemicals, but also products can be applied on those tissue models and get quite accurate predictions for irritation or corrosion.
So in new chemicals, we're looking at individual new chemical substances, not typically mixtures. Maybe the closer thing to mixtures that we look at are new chemical substances with variable composition. For those, we would apply this framework.
Okay, great. Another question. If I understand correctly, the in vitro assays measure cell death as an outcome. This seems an extreme outcome in the case of skin irritation and would not capture irritation associated with activation of tissue-resident immune cells. Is EPA considering how to capture irritants associated with activation of innate and tissue-resident immune cells? Or maybe I can add potential cytokine readouts to that.
So for new chemicals, if we get a result of non-irritating, we're going to go with that, but it might be a little bit different for OPP. And Hans was part of a paper on that, so I'll let him speak to that side of it.
Certainly these models are well characterized for their ability. Thanks, Christy, for also launching on the point of the cytokines. These tissue models do reflect essentially the same type of cytokines that we see in native skin. And, there's quite a bit of work that's been done 20, 30 years now in the personal care and household products arena to look at evaluating for cytokine release in tissue models where perhaps cells death by MTT alone was not sufficient to identify perhaps some mild irritants. And so quite a bit of work has already been done there, published in the personal care arena. And that's an area that we think we can also apply, particularly for the poor category EPA systems where the mild irritants may also be a discriminating factor, which is not part of the new chemicals TSCA categorization scheme.
Great, thank you. There is a specific question, Rachel, on the decision framework. They're asking what you mean by in vivo studies. Are you referring to animal or human studies?
I'm referring to rabbit studies.
Okay, thank you. All right, a couple more questions here. Since skin irritation and corrosion generally increase, I guess, get more severe with concentration, is there a mechanism or guideline for classifying chemicals based on their concentration?
Generally, we adhere to what's in the test guidelines for these and the concentrations that they're recommended to be tested at. I don't have any further comments beyond that. Hans?
I would always recommend I mean, that's a pretty broad question, and I appreciate the logic behind it to avoid testing, but I would probably recommend, if in doubt, test the formulations. And, oftentimes what we do as a contract research organization working with clients, we often look at their specific chemistries where concentration may be very relevant to a specific chemistry and build some database there for them using these models to test a range of concentrations. And then they can use those data internally to build their own prediction models there to determine the likelihood for corrosive or irritant effects before then moving on to some downstream regulatory testing.
Great. We have sort of a statement that the formulations are not working greatly in the 3D skin irritation corrosion tests. And that's not surprising as the validation was on chemicals and not mixtures. You guys might want to add more context to that statement, but there is work ongoing to build suitable tests. How would EPA view these adjustments for mixtures? I guess maybe the general question is, would EPA modify its decision framework if new methods become available?
Yes. Yeah.
Great, thank you. And Hans, anything to add there?
Yeah, I think part of this is we often work with epidermal only models. We're doing corrosion assays with an epidermal only model when reality is that it takes a dermal compartment as well, a full thickness model, to really discriminate the events that happen in vivo between irritants and corrosives. I think maybe anytime that improvements in models are made, obviously one needs to go through the appropriate peer-reviewed publication. We work with perhaps at the OECD level to get understanding of how these improvements might be adopted into a regulatory system. And I think it's up to the regulatory agencies there to then consider how that fits within their framework. That could be a torturous path, but I think maybe in the short run, perhaps also with good literature presentation, one can make a good justification why a modification to a model might be particularly relevant to predicting the irritation or corrosion potential of a chemical and provide that weight of evidence type justification to a regulatory agency.
Okay, great. There's a question about whether there are human disease states that would make the skin more or less susceptible to irritation.
Psoriasis is a classic.
Eczema.
Good.
I said eczema.
Mm-hmm. Yep.
Okay, great. Okay, this is an interesting question. Can you use the epiderm corrosion protocol to determine if a material is corrosive or non-corrosive, and the subcategorization to determine if it's corrosive or irritant? I think what they're referring to the GHS subcategorization of 1A, 1B.
Yeah, if they're referring to the subcategorization, the existing models besides the EpiSkin allow you to subcategorize into either 1A as a corrosive or combined 1B, 1C, which then for labeling is really pretty much a 1B categorization. The methods simply there's not enough data to show discrimination between 1B and 1C for most of the tissue models at this point.
Okay. Under OSHA HASCOM, they prioritize data in this order: human data, then in vivo data, then in vitro data, then structure-activity relationships, and then physical properties. Their question is it right to say that EPA is taking a different approach in this case because they're prioritizing in vitro over in vivo?
We've published on this too. So there's a shift towards really appreciating that the in vitro methods are more relevant to skin irritation and corrosion potential, and they're more reproducible. Um, and that's our justification for taking the NAM studies and prioritizing those over the in vivo data. With regard to human data, we don't see that very often, and we only consider it like on a very case-by-case basis.
Okay, great. Thanks, Rachel. So the last question that I see here, the last relevant question is when there is a discrepancy between test results, for example, an in vitro and in vivo assay, can the in vivo results be considered more reliable? And they bring up the question about animals have an intact immune system where there might be repair mechanisms that are absent in vitro systems, which can affect recovery or injury progression. Hans mentioned this briefly, but could this point be discussed again?
Yeah, I can take that one first. So in new chemicals, the sort of collection of data that we will use for any given new chemical substance to assess for skin irritation and corrosion potential can vary quite a bit. And we'll commonly see conflicting things in that data. If we are in a situation where the NAMS data are saying that something is corrosive, but there's in vivo data that repeatedly shows that something is irritating so that it's reversible, we would potentially use that in vivo data to supersede the in vitro data. But it would really need to be convincing.
Okay, thank you. Hans, anything to add about the in vitro systems and how reliable they can be?
I would only add that the early validation of in vitro systems were almost categorically designed to be protective. Another word for that would be overpredictive. And that I think is the case for the corrosion assay, that there was just no tolerance for any materials to be underpredicted. So it was always compared against the in vivo data. In vivo, so the rapid test already is quite conservative, if you will, protective relative to human skin. And so there's sort of a built-in likelihood that the reconstructed tissue models as well as the cross-techs could overpredict into corrosive category that may or may not be human relevant, may be non-corrosive in humans. So there's a good possibility that the in vivo data might actually be not corrosive for some materials that the in vitro tools predict to be corrosive. I can't speak further to any regulatory application, but I think anytime these type of things happen, these are transformed rapidly into a research project to understand the mechanisms, understand the causes for the discrepancies, and those projects can be very varied depending on any of each example situations.
Okay. Well, we've had a lot of questions, so really appreciate both of you tackling all of those, and appreciate those of you on the phone who've asked all these questions and who listened to our presentations today. Both Hans and Rachel, thanks so much for joining the webinar and sharing your work with us. And thanks to my co-hosts for helping to put on this webinar as this entire series of webinars. We hope it's been useful to you. Please feel free to suggest additional topics, and we will come back with another one in a couple of months. In the meantime, I hope you all take care, and let me remind you that the slides and recording of the webinar will be posted on the PETA Science Consortium website a few days from now. Thanks again, everyone, and have a great rest of your week.